Hello, everyone, and welcome to today's webinar, Wastewater Surveillance, Expanding Microbes of Interest Monitoring Beyond SARS-CoV-2. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Ceres Nanosciences. To learn more about our sponsor, please visit ceresnano.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. I would now like to welcome our speakers. Dr. Dilip Abraham, Associate Professor, Christian Medical College, Vellore, and Pat Acer, Field Application Scientist, Ceres Nanosciences. For complete biographies on our speakers, please visit that biography tab at the left of your screen. Pat, Dilip, welcome. Pat, you may now begin your presentation.
Thank you for the introduction. Hi everyone, my name is Pat Acer, and today I'm going to be introducing Nanotrap microbiome particles for capturing and concentrating microbes from environmental samples. I work at a company called Ceres Nanosciences. We were founded back in 2008 in Manassas, Virginia, based on Nanotrap particle technology. Our hydrogel particles capture and concentrate microbes in a variety of matrices, and our work has been supported by numerous grants over the years, with our technology being featured in over 75 publications currently. The Nanotrap particle technology is based on histology dyes, which are an established technology that do a good job at binding defined cell types as well as cellular components. Pathologists today continue to use this staining technique to evaluate cells to potentially make a diagnosis such as cancer. Specifically with the Nanotrap particles, we attach histology dyes and other affinity baits to our hydrogel particles to bind specific classes of analytes. Analytes bind to the hydrogel particle, which is a porous material, allowing for rapid diffusion of analytes from solution. Also, we can add additional functionalities to the Nanotrap particles. For example, our current products have a magnetic functionality, so we can use magnets to easily pull them out of solution. Nanotrap particles can bind a variety of organisms, including viruses, bacteria, yeast, and parasites, from many different sample types, ranging from municipal or aircraft wastewater to livestock waste. In my time with Ceres, I've actually developed a few protocols for some rather adventurous sample types. The Nanotrap particles are extremely versatile in that they can be integrated into diverse workflows, whether that is a mobile laboratory on wheels or a resource-constrained laboratory with limited access to electricity or refrigeration. With help from the NIH, we have established 21 wastewater-based epidemiology centers of excellence. These sites have provided wastewater testing services for hundreds of locations in more than 100 million people across many states and territories in the United States. Furthermore, our technology can be found globally and is currently used in Africa, the EU, Southeast Asia, and more. So if you're an interested international customer, Ceres is the point of contact for all technical questions and can help you find the correct purchasing path. Our technology is user-friendly. It easily fits into the target concentration step of workflows and offers reliable analyte concentration. Traditional methods like PEG or aluminum hydroxide-based precipitation or filtration methods tend to be laborious with higher input volumes. They can be prone to user error and generally struggle to scale. The Nanotrap technology nicely integrates into high throughput workflows and is compatible with various sample volumes, extraction kits, and detection platforms. If you'd like more information or assistance in method development, please feel free to reach out to me at my email at the bottom of this slide. I will now hand it over to Dilip, who will present on his experience with Nanotrap microbiome particles in wastewater, comparing our concentration method to other established methods across several different pathogens.
Thanks, Pat. Hello, everyone. Great to be here with you today. My name is Dilip Abraham. I'm a microbiologist working at the Wellcome Trust Research Lab at Christian Medical College, Vellore. It is a private, non-profit, charitable medical school in the south of India. And I'm here today to share with you some of the work that we have carried out in detection of multiple pathogens from wastewater, and how we validated our sample processing methods to detect this. Right. So the initial few slides I'll be providing a context for the study, trying to make you understand why we are interested in wastewater surveillance and what we have used it over the years. So this is the study setting, the study area. You can see on the map on the left, the panel on the left, the map of India. So Tamil Nadu is the province that we are in. It is the southernmost state of India, and the colored district, the yellow district, is Vellore. It is a district in the north of Tamil Nadu, and so the wastewater surveillance occurs across 24 wards of Vellore city. Wards is just an administrative unit that we use here in India, and it's spread over 16.25 kilometers square, with a catchment population of 200,000, and the average population density is around 26,500 per kilometer square. This particular population has been subjected to a demographic health surveillance system by Christian Medical College for past several years. So this particular population is censused by the public health department in the community health department in Christian Medical College, Vellore. And so they are geo-referenced, and we also have data on the sewage drains and the water drains. So I started work on wastewater surveillance in 2019. As a matter of fact, our department has been carrying out wastewater surveillance for many years before. Initially, we started out with hepatitis A and rotavirus. The current iteration of wastewater surveillance is basically we started out in the context of polio and salmonella typhi. So this particular study was carried out in 2018 to 2020. Basically, the idea of this particular study is, can we detect these polioviruses, salmonella typhi, and AMR genes in the wastewater using conventional PCR. So it's basically a pilot study in a limited sampling set, and we found out that we could detect it, and and you can see the maps where the polioviruses were detected, and we also managed to sequence the polioviruses in collaboration with Imperial College London, and we are also able to detect the TTR gene of salmonella typhi. Now, the next study in 2020 to 2021, this is a study in which I was involved. Now that we know that we can detect these targets by molecular detection in the wastewater, we wanted to go do this in a more systematic level. We want to answer questions like, what exactly do you mean by a positive sample for something like bacteria, right? Is it just a single target, or do you take multiple targets to define that as a positive sample? Which is the most appropriate site for sampling for doing, is a grab sampling better, or is a trap sample better, and what is the optimum catchment area in which we should be doing this. So this particular study was designed to try to attempt to answer these questions. So we had around 40 sampling locations, which is mapped into catchment areas that are upstream of it, and we did both trap sampling by MOSAR and grab sampling with membrane filtration in this particular study. But I think you all know what happened in 2020 and 2021. The pandemic struck, and what we were trying to do was basically trying to validate our wastewater detection by carrying out a parallel clinical surveillance in those particular catchment areas. Now, because of the pandemic, we are not able to access the population because of the highly restrictive lockdowns. So we were stuck. So by the middle of 2021, things had kind of opened up. So by October 2021, we did a sero survey, and we utilized a novel technique using hemolyzing E IgG which is developed by Dr. Jason Andrews in Stanford and Dr. Richard Charles at Massachusetts and Dr. Christian Enjoy. And basically, by doing a cross-sectional point prevalence sero survey, we are able to extrapolate the sero incidence in each catchment over the last 1 year. And this has been published by Christian Enjoy's paper. And these are the results of that we can see. You can see the sero incidence in each particular catchment varied all the way from 5 to around 25 per 100 person years. The overall sero incidence was 10.4%. Now, what we wanted to do was to, we are not actually interested in sero incidence, we wanted to validate our wastewater detection. So what we tried to do was do a regression analysis to kind of correlate or to figure out an association between the sero incidence in each catchment and the wastewater detection at that catchment over the last 1 year. And we carried out negative binomial regression, and we found out, yeah, the sero incidence was associated with wastewater positivity. And so you can see the association for membrane filtration alone, for mood swab, and the overall rate. Right. So, and in this particular study, the sero incidence demonstrated higher association with membrane filtration, even though mood swab was twice as much as positive as membrane filtration. Right. So that is the results that we got here. Now, coming to the current study that I'll be talking about. Now, once this was done, because we had multiple limitations, we couldn't carry out clinical surveillance. We could only carry out sero incidences. We wanted to try and do clinical surveillance also. And in this particular iteration, we also was interested in trying to test for multiple targets, including in addition to Salmonella typhi, Vibrio cholerae, SARS-CoV-2, and hepatitis E. And the primary objective of this study was to figure out whether we can develop one single method and validate it, so that for one single grab sample method and one extraction and multiple PCRs, can we detect all these targets at once, right? Because if we want to transfer this methodology to other labs in India, nobody wants to do multiple methods for multiple targets. So that is the aim. And initially, we started out by looking at these particular qPCR targets, 3 targets for Typhi. We had actually 6 targets for cholera, 2 for SARS-CoV-2, and we zeroed down on 1 for hepatitis E. The sample processing methods, we went with mood swab, and grab sample methods, we are trying to test out membrane filtration, PEG, skim milk protocol, and bag mediated filtration method. So this was our initial plan. So we started out by carrying out spiking studies. So we use heat inactivated SARS-CoV-2 to spike wastewater samples in order to detect SARS-CoV-2. We had a TY2 control strain for Salmonella typhi and a clinically isolated strain, O139 L2 strain for Vibrio cholerae. And we spiked 3 different samples of the same grab sample in different concentrations of spiking, and we calculated the recovery as depicted, basically because each particular methodology would sample different volumes of the wastewater, right? So it is important for us to do the volumetric adjustment to get the actual recovery rates. So we first did the spiking studies for SARS-CoV-2, and you can see the preliminary results. We did it for both wastewater. We were trying to figure out which is the best sample matrix also. So we did it for both wastewater and sludge. We found out that the skim milk flocculation technique provided the best recovery for wastewater and the direct extraction for sludge. If you look at the numbers, it was evident that sludge was a much inferior sample matrix in order to recover the SARS-CoV-2. So that is what we observed. So we thought, okay, we had our sample processing method down, but unfortunately, then what happened was molecular lab grade skim milk became unavailable in India. So the Indian government banned the import of skim milk because we are only allowed to use skim milk that is manufactured within India. And the problem is molecular lab grade skim milk. Skim milk, if you those of you working in the lab will know that skim milk is used for mainly as a blocking agent in ELISAs. So which is fine, we still use that procured from Indian manufacturers. But when you're looking at molecular lab grade skim milk, it is very it's just not available, and I was not willing to try that out. So we had to move to other modalities. So that is when we heard about from Dr. Scott Meshke, we heard about, and Dr. Mamit Anyuchi, we heard about the nanoparticles, that's hydrogel particles. So we reached out to Series Nanosciences, and this is a final plan. We replaced bag mediated filtration method and skim milk method with Nanotrap A and Nanotrap B. And we wanted to basically do a head-to-head comparison of these sample processing methods to figure out which is the most optimal graph sampling method in order to detect all these targets. So now I'll move on to the results from the study. Yeah, so these are the results from the spiking study for SARS-CoV-2. The box plot on the left is for N1, the box plot on the right is for N2, and the green box is depicts the recovery for Nanotrap A. And as you can see, it is pretty evident that Nanotrap A, especially for N1, is much more efficient than all the other methods to recover SARS-CoV-2 from wastewater. That's what we found out in this particular spiking study, and this difference was statistically significant also. So we are pretty happy about that. By this time, we had been using the Nanotrap method in the lab for about 2 to 3 months, and we are all very happy with it because it's very easy to do. Now coming to the spiking studies with Salmonella typhi, here you can see it's not that evident, right? Because with all these methods, if you look at the box plots, they all kind of overlap. So there is no significant, statistically significant method that's better. But again, the lime green, the kind of green box plot depicts Nanotrap A, the blue is Nanotrap B, the purple on the right is PEG, and the red on the left is membrane filtration. And again, you can see that Nanotrap A overperformed the other methods for recovery, even though it's not statistically significant. The median recovery was much better. This is for all three targets that we use for Typhi. This is the pan Salmonella TTR, the TVIB gene, and the STAR gene. Right. So here we have the results of the spiking study for Vibrio cholerae, right? And here the results are a little more different. For the panel on the top, the graph on the top left is the recovery for OMPW, which is a pan Vibrio target. And here, regardless of whatever target it is, you can see it's not statistically significant. All these methods perform pretty comparably well. But even then, you can see for OMPW, Nanotrap A is better. Interestingly, the toxigenic target CTXA, which is on the upper right box plot, you can see that none of the methods detected CTXA except Nanotrap B, which was kind of puzzling for us because we were by this time we were hoping to zero down on Nanotrap A. But for the other targets, basically O139 and TCPA LTOR, we found that Nanotrap A kind of it performed at least visually. You can see that the recoveries are kind of better. So those are the results from the spiking studies. This is now we carried out head-to-head comparisons for 6 months of sampling. That is in this particular study, that's like 50 sites and sampling graph samples twice a month, right? So that is around 600 samples, and here you can see the percentage positivity. So basically, these are unspiked samples, and here you can see a much more much more complicated scenario. Nanotrap A is what we wanted to use, and none of these methods are statistically significant or statistically different from each other. But we narrowed down on PEG 8000 precipitation and Nanotrap A as the two methods that perform consistently across all targets. But Nanotrap B is also pretty good. But PEG precipitation, Nanotrap A was what we did. Now, the membrane filtration, we kind of abandoned that method because the issue is the wastewater, the sample matrices that we find in India, they are highly viscous. So we will be left filtering the sample for the entire day, and so which is not possible. It might take 4 to 5 hours to filter one particular sample. So we have to have a defined cutoff. Let's say we'll only filter the sample for, let's say, around 2 hours or 3 hours. In which case, the amount of sample that is filtered is varies a lot. So we were not happy with that. Now, we also carried out sequencing from the so these are basically the results of the 6 months of sampling from the wastewater. These are not positives. These are just the y-axis is just the genome copies per microliter, and this is just a summary of what I showed you. So the wastewater samples are subjected to all 4 graph sample methods and moved for 6 months. And like I said, the Nanotrap A method and the PEG precipitation method show similar positivity for all targets, and the difference was not significant. So, as I was saying, we carried out sequencing of SARS-CoV-2 from wastewater, and these two box plots shows the mean coverage across all different methods and the mean read depth across different methods. And again, these are not statistically significant, but Nanotrap A and PEG, they were kind of comparable for SARS-CoV-2. So now the question is, like, in our minds, where do we go ahead with Nanotrap A or do we use PEG? Basically, if you just to remind you, our aim was to narrow down on one or two graph sampling methods that is scalable and transferable to other labs in India. So CMC Vellore is a highly specialized. Our lab is highly specialized for ES. We have been doing for multiple years. But is this? Can we write a SOP or a protocol that other labs in India with access, which may not have access to this specialized equipment, will they be able to do this? So we try to look at the pros and cons of each method, because these two methods, the Nanotrap A and PEG, were the ones that were, in our opinion, were performing the best. So if you look at it, the first point is that Nanotrap A is very easy to carry out, and PEG 8000 precipitation is more tedious, right? So this is kind of the the tiebreaker that allowed us to say that we are going with Nanotrap A. The reason is for Nanotrap A, the sampling volume is around 40 ml, and PEG 8000 we use around 250 ml. That is, I believe, that's the reason why we get the PEG is the recovery efficiencies and the recovery of these targets are comparable to Nanotrap A, because we are using at least 2 times the volume. More than 2 times the volume. But the problem with using higher volume is that the methodology becomes more tedious. You need to purchase a megafuge for sample centrifugation in order to centrifuge these large amounts of sample. But Nanotrap A, you don't need to buy that. All you need is 2 magnetic racks and the Nanotrap reagents, right? And the other advantage of Nanotrap A is, and PEG, is that these both have comparable results for sequencing when compared with Nanotrap B or membrane filtration, in our opinion. Now, what are the disadvantages of the Nanotrap particles? The per sample cost is higher, right? But this sample cost is offset by the because in PEG we need to get a megafuge, or Nanotrap we don't need to get that particular equipment. But when you scale it up, the cost becomes kind of similar between both. The other disadvantage of Nanotrap A is that the pellets need to be extracted within 24 hours. Right? It cannot be frozen down. It has to be refrigerated and extracted within 24 hours. For PEG, the pellets that we get can be saved and can be frozen at minus 80 degrees and extracted in batches. So that is something that we don't have to do real-time extraction. Just let's say in one particular day we only have sampling from 2 or 3 sites. We can save those pellets and do it later. Now, one of the things that we are paranoid about after the pandemic is the supply chain issues. Right now, it has been like 2 years since the pandemic, and things are getting much better. But in 2023, when we did this study, the supply chain was not good at all. And but I must at this point, really compliment the sales team in Series Nanosciences. They have been absolutely great. The pleasure to work with. But if you recall the story with skim milk, the Indian government just banned the import of skim milk from external sources. So the supply chain issues in India depends on lots of external factors. So that is something that we are still concerned about. But however, we are still continuing to use Nanotrap A. And in case the supply chain fails, then we have the alternative of PEG 8000, which is kind of available from multiple sources. Now, that concludes the head-to-head comparison of methods. So in the next few slides, I'll just show a few results from the 1 year of sampling that we did. So these are the results of SARS-CoV-2 wastewater surveillance, and you can see the panel on the left shows it's like a time series plot. The red area is the Nanotrap A particles, and the blue area is MooSwab over 1 year. And you can see the spikes in detection during the months of January to May 2023, followed by a peak in the monsoon season here. That's August to November 2023, right? And the panel on the left is N1, the panel on the right is N2. And the figure on the right is the grid map plot, in which you can see the spatial resolution. So at each point of sampling, you can see that even there is a spatial variation. So SARS-CoV-2 is not detected in the wastewater from all the areas, but only in specific catchments. That's actually kind of correlating with the population density in those areas. Now, in order to validate this, we wanted to get some data from the government on SARS-CoV-2 case data. But in 2023, nobody in India was interested in SARS-CoV-2 cases. People are just refusing to report it because the lockdowns were fresh in memory, right? So in this particular, when we attempted to get the data, but in this particular year, there's only 7, or from this catchment area, there are only 1 to 2 cases of SARS-CoV-2 that was actually detected, which we know is false, right? There is a concurrent SARS-CoV-2 vaccine trial going on, and in 2023, and we are trying to access data from that in order to correlate with detection in the wastewater to case data. Another way of validation is trying to sequence the wastewater that were positive for SARS-CoV-2, and we did that. We sequenced the wastewater using the ARTIC protocol, and the demixing of the samples were carried out using the Freya pipeline. And here, in each particular panel, you can see this is separated out by the SARS-CoV-2 variant, and you can see variants like XBB, XBB 1.5, and XBB 2.3. They kind of correlate with each other with the influenza and SARS-CoV-2 seasons. Especially if you look at the green line, that is CH 1.1. This particular variant was detected the first detected in the world by September 2023, detected in India only in November and December 2023. But you can see in the wastewater, it was detected at September 2023. So we kind of were able to demonstrate that in the wastewater, these variants show up much earlier than that they would be detected in the cases. Now, coming to the results of the Salmonella Typhi wastewater surveillance, the plot on the left is a time series plot over 1 year. The red depicts it's over 6 months. The red depicts the case data, the blue is the Nanotrap A data, and the green is the MooSwab data. And you can see, when the case data spikes, you can see a corresponding spike in the MooSwab and the Nanotrap A detection of Salmonella Typhi. And if you look at the autocorrelation plot, we found that a lag of 5 days between the wastewater positivity and clinical incidence is significant. Now, when we carried out the same negative binomial regression, we found out that Nanotrap A, the incidence rate ratio of Nanotrap A positivity to the case data was around 1.85. It was 2.03 for MooSwab. We also carried out multivariable negative binomial regression, and we found that there's a significant interaction effect between the population in the particular catchment and the positivity, which is kind of what we expect. Now, these are the results of Vibrio cholerae. Now, we were interested in Vibrio cholerae because cholera strikes these peri-urban and rural areas in Vellore, and the issue with that is because these particular areas do not have good access to healthcare. By the time the patient is diagnosed as cholera, by the time he or she, usually these are children, by the time they get to the hospital, they fall into a very serious state, and there are even fatalities. So, which is why we thought And the issue is, again, people are very hesitant in reporting cholera and to the public health administrators in Vellore, and they are also not receptive to these cases. So, which is why we are interested in looking at cholera. Here again, we try to access case data um, from the authorities, but again, the case data is very, very sparse. However, if you look at the spatial map, and if you also look at the time series on the left, you can see there is a spatial and temporal variation in cholera. Usually, it increases during the post-monsoon time points, and that's what we see. Right. So I would like to end with some of the challenges that we faced and some of the things that we are thinking about how to take this further. So definitely we need to validate all methods at our own sites. If you go back to the spiking studies, if you look at spiking studies that were done abroad when I say abroad in the United States or in Europe, you can see a much higher recovery efficiency of around 20%. But our recovery efficiency for SARS-CoV-2 is around 4%. So definitely the sample metrics is very, very different. So we need to validate the method at their own site. There are lots of move swabs. We need to manage time when we scale up. Again, reagent procurement was a headache in 2023. Now it's much better. And then another thing we noticed that when we after extracting the DNA, if you had to go back and when we did the PCR, if you want to go back to the same DNA, the freeze-thaw DNA, there is a reduction in detection by an order of magnitude, right? So the CT value increases by around 3 when we do a freeze-thaw. So this is something that we need to be careful about. Now the most important challenge is defining what is a positive sample. Is it just for SARS-CoV-2? Is it just N1 or is it N1 and N2? For Salmonella Typhi, should we go with? So in this particular study, we went with if all 3 targets are positive, only then do we define it that it's Salmonella Typhi is positive. Similarly for Vibrio, we defined as only if OMPW and CTXA is positive, only then is that particular sample positive for Vibrio, right? So this particular definition should be validated either by sequencing from the wastewater or by kind of correlation with the clinical cases or clinical surveillance. So that's kind of the major challenge of wastewater surveillance, because we need to get public health personnel to believe the data, right? Now, obviously, the other silver bullet is like, how do we normalize? Is an increase in detection due to an increase in burden, or is it due to just an increased amount of feces that is there in the graph samples at that particular point? We try to carry out normalization with HF183 and graphage, but it's not still apparent how to actually normalize it. Or do we just use flow rate? We also have measured flow rate, but this is work in progress, and there is not consensus in the community on how exactly you normalize graph samples. The other issue, obviously the biggest issue, is how to communicate with public health personnel on how to use this data. So if you go to a public health person in India, they'll say, see, we have all these positives, and they'll say, so what? What do you want us to do about it, right? So we need to present them with actionable data that is validated either by sequencing or by case data, and we need to have some plan of action. There should be some consensus in the public health community, like if there is an uptick of cases for influenza or SARS-CoV-2 or typhoid that we see in the wastewater, what do we do then, especially in developing countries or resource-limited settings? Yeah, so that ends my presentation. We are now open for questions. Thank you.
Thank you, Dr. Abraham, for that excellent presentation, and welcome back, Pat. It is now time for our live Q&A portion of our webinar. Now, to our audience, if you haven't submitted any questions yet, please do so now. Type them into the Ask a Question box and click Submit, and we will answer as many of your questions as we have time for. So, gentlemen, we have so many questions that have already come in, so I'm just going to dive in. So, Dr. Abraham, let's start with this one. What are the major challenges when carrying out environmental surveillance in a low to middle income country?
Yeah. Hi, Christy. Thank you for the question. Well, obviously, in any resource limited setting, the main challenge is basically resources. We are carrying out wastewater surveillance because it's supposed to be much more cost effective than doing a clinical surveillance. But even so, doing molecular testing is quite expensive, and you need the infrastructure and the funds for that. Supply chain, like I said in the last slide, it used to be much worse last year. Now things are much better, but even then, supply chain is still an issue for things like probes that we use for PCR. Once they order them, they have to be manufactured outside India and then imported. So that leads to delay in the supply. Um, Much more important question is like, how do we communicate effectively with public health personnel, and how do we make this data actionable? I think that is a major challenge. If you go to say, okay, we have an increase in detection of dengue from the wastewater, I mean, what does that actually mean? Should they take it seriously or not? Right? Even during the SARS-CoV-2 times in India, I was talking to a public health person, and we're talking about wastewater, and he or she was telling, okay, suppose we see a increase in detection of SARS-CoV-2 from the wastewater, what are we supposed to do? People are locked down anyway. So that is the major challenge. How do you make this data actionable, and how do you manufacture consensus on what to do when there is a uptick of detection in the wastewater?
Thank you. And Dr. Abraham, this question's for you as well. What are the prospects for sustainability of ongoing environmental surveillance in low to middle income countries?
Yeah, so sustainability is the issue, right? So I think for the multi-pathogen grant, one of the aims was to devise methods that are feasible to do in India and scalable. So number one, we should be practical. I mean, we are not. I mean, it's not feasible to do ultra centrifugation or buy this highly specialized equipment if you want to do it on a large scale in different places in India, right? So we need to be practical, and we need to be practical about what we are capable of doing. So that's the first thing, and we need to use methods that are accessible and feasible to be used for example, move swabs. Another example I would say is composite sampling, right? If you look at studies from outside India, many people, they use composite samplers, automated composite samplers in STPs or sewage treatment plants. In India, we tried to do that, but it's just not possible because these samplers are very expensive, and there's issues with safety and issues with power, access to power. So we need to match the method to what we want to detect, to the location in which we are. The other thing what we might need to match is you need to fit the tool of wastewater to the use case, right? So, for example, if you're looking at something like a sentinel surveillance for SARS-CoV-2 or monkeypox, then the number, the sampling frequency and the sampling sites need to be modified to reflect that. If you're only interested in the burden of some endemic disease like typhoid, then you don't need to sample that often, right? So you need to make these trade-offs, and you need to think about how to most effectively leverage this wastewater surveillance for public health. Thank you.
Thank you, Dr. Abraham. Pat, I'm going to come over to you for this question, and this one's two parts, but they go together here. What's the difference between Nanotrap A and Nanotrap B? And can you elaborate on the ability to supply Nanotrap particles globally?
Yeah, for sure. So the primary difference between the Nanotrap microbiome A and B particles is actually in the specific affinity baits that are attached to the hydrogel particles. And they have an impact on sort of which microbes each particle can capture. So in practical terms, microbiome A might be better suited for a specific type of microbe, whereas microbiome B may be better suited for another microbe. I will say there is quite a bit of overlap in the microbes that both of these particles can capture. So actually in studies that we've conducted internally, we see that the combination of microbiome A and B particles can really enhance sensitivity of an assay. The second part of your question, so Series ships direct next business day for all of our Nanotrap particles, and we work closely with global customers to ensure smooth customs clearance.
Excellent. Thank you so much. And Pat, I'm going to stick with you on this next one. Does the presence of salmonella or bacteria increase the likelihood of finding viral infections in wastewater? If yes, how can you account for this?
Yeah, so I would say the presence of salmonella itself wouldn't always indicate a higher viral load. However, salmonella, along with other bacteria, can oftentimes serve as indicators of the proportion of fecal contamination. So finding salmonella itself could indicate that that specific sample of wastewater has a higher proportion of fecal material. And with that can also come sort of a higher likelihood of viral pathogens being present, and how one could deal with this. Dilip touched on this a little, but would be through normalization. So many groups use different normalization markers to account for that fecal input in a given wastewater sample. Um, However, it's geographically dependent, and finding one that is relatively stable over time tends to be quite important.
Thank you, Pat. And Dr. Abraham, I'll come back to you. In the absence of the co-presence of these infections, do you have any idea how well the Nanotrap A and Nanotrap B perform in concentrating either of the pathogen?
In um, absence of the co-presence, I'm not very sure what this means. I mean, do you mean like if all the other pathogens are not there and only one pathogen is there? I think I would I would probably assume that in the absence of other pathogens, the sensitivity would increase because the nanotrap particles are able to adhere to more particulate matter in the wastewater. That's what I would assume. But regardless of we haven't done co-spiking experiments in which we spiked both SARS-CoV-2 and Vibrio and Typhi. But we did have samples which are positive for both Typhi and Vibrio, right? So I don't think these. I would assume that I mean I don't have the evidence for this. I would assume that sensitivity would decrease, but it does not decrease. I'm pretty sure it does not decrease to high significant level.
Thank you. Now, Dr. Abraham, why the skim milk method? Why was this not taken into consideration any longer in your experiments?
Yeah, so this is not something which we wanted to do. We were very happy to use skim milk because it is easier to do than PEG. The issue with skim milk, like the Indian government in 2023 banned the import of lab grade skim milk that we use for this, right? So the skim milk that is available from India, we use for ELISAs, for blocking agent for ELISAs. And sometimes we can detect, if we use that for PCR, we can detect targets like HF183, stuff like that, which we use to normalize the wastewater. So the problem is which supply do we trust in order to get lab grade skim milk? That would be negative for all DNA targets, because even if you autoclave it or do some kind of you can still might detect some DNA. So at that point, I was not very interested in utilizing skim milk because it is highly organic, and because of the supply chain issues and because of the quality issues, we went with PEG, which is kind of still organic material, but we have used it for a long time, and we are pretty happy with that. And Nanotrap A, of course.
Very good. Thank you. Pat, I'm going to jump back over to you. Can these concentration techniques also be applied to well water, from which many populations across Africa get their water supplies? Well water versus wastewater.
Yeah, I would say for sure. I've actually personally been working with other environmental sample types, mostly tap water, but we do have plans to expand into other drinking water sources as well. I would say we know that the nanotrap particles themselves are definitely compatible in this matrix and do really a good job at binding different bacteria.
Thank you. Dr. Abraham, what could be the possible reasons for the success of Nanotrap B for that one result you showed in your box plot? were all the other where the box plot where all the other results were negative?
Yeah. So one of the things I've learned over the last 2 years in doing wastewater surveillance is that the bacterial targets are an order of magnitude much more complicated than viral targets, right? So viral targets are kind of easier to detect. We know what a positive sample is because if for SARS-CoV-2 if N1 and N2 are positive, then that's a positive. For things like Salmonella typhi and Vibrio, it's very much more difficult. For Salmonella typhi, we had to end up with three different targets that are kind of specific for different targets of Salmonella typhi, and only if all three targets are positive, we counted that as a positive, right? The same way for Vibrio, and and one way you can think about is, for example, Vibrio O139, right? O139 target was positive in around 100 samples in 2023, but the last time that pathogenic O139 was isolated from the patient in Vellore, in this part of the country, was way back in 2008, right? So just because a target is positive for a bacteria in the wastewater does not necessarily mean it's positive for that particular pathogenic bacteria. So just with that context, if talking about CTX-A, why it was better in Nanotrap B, I'm not that. I mean, I'll have to go to Pat because even in their website they look at Nanotrap B as being better for bacterial targets. But for the spiking, we certainly found that CTX-A was picked up in Nanotrap B. But when we looked at the actual samples without spiking, both Nanotrap A and Nanotrap B both picked up CTX-A. But again, Nanotrap B was much better. Actually, we are pretty surprised that Nanotrap A outperformed Nanotrap B for bacterial targets like Salmonella typhi.
Thank you. And Dr. Abraham, which enhancement reagent did you use? Did you do it by hand or use Kingfisher? If so, which program?
Right. So I have had bad experience with automation in India. The problem is, much of these automators are kind of they are made with something with a clinical sample in mind, like blood or stool. The problem with wastewater and things like that is that the sample matrices are very very different. So during the rainy season your sample will be like blood, and during the summer season your sample will be like stool, right? So that because of that, these automated samplers, at least in our setting, tend to get clogged very easily, and they tend to break down, and then you need to wait 2 weeks for the technician to come to your institution in order to fix it. So that's why we were not very interested in automation. Some kind of semi-automation would be good, but again, we require support from basically the companies in order to do that, and such kind of support is not consistent in our part of the country. So all these methods are done manually because manually, even though in the long run it is more expensive because you need to pay technicians or you need to do it yourself, but it is more reliable to do it manually in at least in our setting. Enhancement reagent for Nanotrap, I mean, I think series itself has provides enhancement reagents. I think it's ER3 for Nanotrap A and ER2 for Nanotrap B. Correct me if I'm wrong. So that's what we used in this particular, in the graph sampling methods. We didn't use any other enhancement reagent other than that.
Yeah, so I can add on a little bit there. The enhancement reagent is in part dependent on which extraction kit you're using, and we find that some of our SKUs sort of are more compatible with certain extraction kits compared to others. So we'll make specific recommendations based on a group's specific needs and which kit that they prefer.
Thank you, Pat. And I'd like to direct this next question to you. So if you want to storage the samples before DNA extraction, which method is better? And how long can you store the wastewater samples before DNA extraction for pathogens as Salmonella or Vibrio?
Yeah, for sure. So I guess I'll answer this question two ways. So if the question is referring to raw wastewater, I would say you can store it at +4C for days to maybe even weeks, depending on the abundance of the target in the matrix. We have seen groups freeze the wastewater at -80. I will say that at least with Nanotrap particle concentration, our particles capture intact microbes. So you would expect to likely see a lower recovery of analyte after a freeze-thaw cycle. If that's a super abundant analyte, then maybe it's not that big of a deal. If the question is referring to after the concentration step, when the sample is stored sort of as a lysis buffer, so we've lysed the organisms, then we don't necessarily recommend it. We recommend going fresh into extraction with the lysate, but we have seen that you can freeze the lysis buffer at that point as well.
Thank you very much. All right. Pat, let's have you take this next one. Can wastewater surveillance for antibiotic resistance potentially help in antimicrobial stewardship and making more appropriate infectious disease treatment decisions?
Yeah, I would say for sure by, say a hospital is identifying specific ARGs, subsequently clinicians can choose which antibiotics are more likely to be effective, avoiding those ones that may not work against the bacteria that have conferred resistance. Yeah, I think this could enable different sorts of infection control measures, such as like isolating a certain pocket of patients within a specific building to really prevent the spread of a specific problematic organism.
Excellent. And we have time for a few more questions, so let's squeeze them in here. So we believe this is magnetic based. Why would I need to choose only Nanotrap and not other beads? And Pat, you can answer that one.
For sure. So I would say first, we know that our nanotrap particles are almost universally compatible with any extraction kit, which our competitors can't necessarily say. I would say our cost in general is lower, especially at scale, and the nanotrap particles themselves offer sort of a versatility and flexibility that some of these other particles on the market don't necessarily guarantee, in that our particles can work in a variety of environmental matrices. And, we have a dedicated team here that is happy to validate and develop protocols in sort of any new matrix that we haven't tried yet. So I think initially that is what comes to mind on like why we're better than our competitors, or why a customer may prefer our products over other products.
Very good. Thank you. Dr. Abraham, let's have you take this one. Which volume did you use to concentrate using Nanotrap particles?
So we used 40 ml of the wastewater. That's what we.
Excellent. Thank you. And we're going to wrap with this final question. And Pat, I'll have you close us out with this one. What about the re-emerging Mpox virus and other viruses, polio, hepatitis A, regarding monitoring?
Yeah, so I guess simply put, I'm interpreting this question as like, can the nanotrap microbiome particles capture these targets? The short answer is yes. We've seen that we can capture all of those targets that have been mentioned. The nanotrap particles broadly capture microbes, which is great when sort of there's a new emergent pathogens that groups are like, can we capture this? Oftentimes we find that that pathogen is already there, even if they weren't necessarily looking to sort of capture that in the first place.
Thanks, Pat. Now, before we close, I'd like to give you both an opportunity to say some final comments for our audience. Dr. Abraham, would you like to go first?
Yeah, sure. Thank you all for listening to us. And if anybody is there from India, and we are always at Vellore, we are always open to collaborating with you. Most of the protocols that we use here that I explained are published on protocols.io, and so you can reference that, or you can just email me and get in touch with me if you have any doubts or if you want to collaborate. Thank you for listening.
Yeah, just going off that, thank you everyone for being here. If you have any questions about do nanotrap particles work in this matrix or would they work well in my workflow, I did have my email on that initial slide, so feel free to shoot me an email. I'm always happy to help answer questions and collaborate to try and integrate sort of our technology into your workflow.
Thank you, Pat. And I just put that email right in front of our audience so they have that handy. But I do want to thank you both again for your time and your very important research in wastewater surveillance. I do want to thank LabRoots and our sponsor series, Nanosciences, for underwriting today's educational webcast. Before we go, I do want to thank our audience also for their interesting questions. We had quite a few questions we were unable to answer due to time constraints, but additionally, those questions that are submitted during the on-demand period as well, they will be addressed by our speakers via the email address you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues and your friends who may have missed today's live event. Thank you again for joining us. Until next time, have a great day.